The separation of colored substances from a mixture of pigments. Many naturally occurring dyes and inks contain different pigments. For example, black ink may contain red, yellow, and blue pigments, while the green leaves contain chlorophyll, that is the green coloring matter, and xanthophyll, which is yellow in color. In order to separate these pigments, we employ a method known as chromatography. The diagram here shows an example of paper chromatography. In a paper chromatography, two properties are very important. The first property is solubility, and the second is adsorption. Now the ink is placed on the baseline, and then the paper is dipped into the solvent. At the beginning of the experiment, the solvent is absorbed by the paper, and once it reaches the ink, it dissolves part of the ink pigments in different categories and carries them with it upwards. At the end of the experiment, you'll find that different pigments have moved different distances from the baseline, showing that they have different solubilities, different adsorption properties. The yellow pigment, for example, moves the farthest from the baseline, which is right down here. Because the yellow pigment is the most soluble and least adsorbed. Pigment. The level where the farthest point the water reaches, is known as the solvent front, while the paper material where adsorption takes place is known as the adsorbent material or chromatogram. An example of paper chromatography that can be carried out is shown in the picture below, where we have different colored of inks. At the beginning of the experiment is the start before the experiment, and at the end of the experiment the chromatogram was removed from the solvent and the results were obtained as follows. You may observe that the red pigment or the red ink contains yellow and red pigments. Blue ink contains blue and green pigments. Green pigment contains green and blue pigments. Brown pigment contains black, red, and yellow, while the black contains red, blue, green, and yellow pigments. These pigments move different distances from the baseline, which is this, because of the different properties of solubility and adsorption. Chromatography is very important and it has various uses. Firstly, it is used to identify illegal substances such as steroids in blood and urine samples. This mostly applies in sports to prevent doping. It is also used to identify harmful substances in cosmetic products so that the manufacturers do not include any harmful substances that may cause damage to human beings. It can also be used to identify contaminants in food and beverages in the food industry. In the pharmaceutical world, it is used to test for purity of drugs. The diagram shows how chromatography can be used to identify illegal substances in blood and urine samples.